I'll do more or less. I don't think the title is a bit a fancy title. I will go through different things. As you know, I don't think that TDP-43 needs to be presented to this audience. Anyway, just to remember that have a lot of cellular functions, some in the nucleus, some after shuttling to the cytoplasm. And I am going to tell you a little bit about this function, that is the one that through which TDP-43 regulates its own cellular levels, and a little bit about the aggregation of TDP and methods to mimic what we see and measure the consequences. As you all know, the first function of TDP-43, the one that was well documented well before it became a famous protein, was this function in the exon 9 of TDP-43. And there, in that system, we discovered that that was the protein binding here to the 3' splice site of TDP-43. And as Jerny showed yesterday, being in the 3' splice site has an inhibitory effect. And seems that that inhibitory effect was mediated by interactions with other HNRMPs like A1 and A2. Obviously, this protein, we realized at that time that was a classical HNRMP. It was supposed to be a DNA binding protein, but this actually has the structure of an HNRMP with RRM domains, nuclear localization signal, glycine-rich C-terminal domain, through which all the interactions are mediated. A very interesting feature was that it's very, very well conserved, not only in structure, but also in function through evolution. So Drosophila protein can be exchanged with a human protein and in all at least the splicing function that we study. And you see here one example. I mean, in this system, if you mutate the RNA binding ability, then you lose the possibility of recovering inhibition of splicing. No? Here you have the CFTR exon 9 in the two forms. You do an S size against TDP, and then you get recover of full inclusion. You transfect wild type, you get back inhibition. You transfect the Drosophila, and you get back inhibition. And you transfer a mutant that cannot do RNA binding, and you don't get back inhibition. So RNA binding was essential for its function, at least in splicing. Then, to continue studying the properties of the enzyme, we made stable cell lines with a flip system where we expressed transgenic TDP-43. And this was our surprise. The first thing that we saw was that when you induce the transgene, the endogenous one disappears. So there was an effect of the transgenic TDP-43 on the expression of the endogenous. Thus, for us, that we are RNA molecular biologists, was very interesting, almost as interesting as the role in neurodegeneration, if not more. And we started to study that process. It's quite dramatic. I mean, you can see here, 24 or 72 hours after induction of the transgene expression, the endogenous protein disappears. If there is an RNA mutant, the transgene, then there is no autoregulation. TDP-43 has two messenger RNAs, a short one, 2.8, and a long one, 4.2. And both disappear with the in excess TDP-43. So the disappearance is quite dramatic, and obviously there is no such a disappearance when you use a RNA binding mutant. You can see here a little bit there is remains more of the longer one than the shorter one. We still don't have an explanation for that. We studied a little bit, and I'm going to go fast because I know that this may not be of general interest for neurodegeneration, although I think it is. We studied the mechanism. We made the hybrid of GFP with the 3' UTR of TDP that contains this area here that we call intron 7, and when in collaboration with Int'l, we have found a lot of TDP-43 binding sites. So after many manipulations of this, we realized that deleting that area, it will abolish self-regulation. In this case, what I am showing here is the effect of overexpression. This will be the equivalent to the long messenger, and normally most of it in the mini-gene is finishing in the poly A1, that is this one. When you have a transgene overexpression, that TDP binds here and induces splicing of this in the 3' UTR, and this is the short version. From here, jump the intron to this poly A2 site. If you mutate the splice sites, then there is no regulation and no splicing, obviously. You can see that at the level of the protein is quite well conserved. And let's say represent very well the change. Why this amount of messenger is not producing protein, we'll see it in a moment. The endogenous TDP-43 of the cell behaves in the same way, producing this same messenger here and dropping the amount of protein. We thought at the beginning that the poly A2 usage was the problem. I mean, that was an inefficient poly A2 and there was some problem. Actually, then we made this cDNA that was as the splice product, as the one that we named poly A2 in the previous slide. This splice product expresses very well in the cell, it has no problem, makes a lot of protein, and is not obviously self-regulated. To see if there was a difference between the original mini-gene and the cDNA in the amount of messenger, we co-transfected both. This normal cDNA, no overexpression of TDP. This is the cDNA, and this is both together. As you can see, there are fairly equivalent amounts, and of course, they produce fairly equivalent amount of protein. This means that poly A2 messenger are perfectly translatable and have no problem in going out of the nucleus, and there is something else. What is the something else? We look at it again, improving the splice sites. So saying we are going to force this to do always splicing in the presence of an excess TDP-43 or in the presence of normal TDP-43. And in fact, if you improve the splice sites, we call it super splice sites, you get perfect constitutive splicing and production of the poly A2. But in contrast with what you see with the cDNA, this is the same messenger as the cDNA, but you have much lower production and much lower production of protein. So this type of process, if you synthesize in vitro the cDNA, perfect, works wonderfully. If you synthesize it in the cell through splicing, it doesn't. So the idea is that something during the splicing process marks this RNA for degradation, nuclear retention, or whatever it is. Our idea, and well, we have further evidence that there is a difference because these are fish RNA, normal construct with a GFP 3' UTR. the super splice construct and the cDNA construct. You can see that the messenger in the case of the cDNA is all in the cytoplasm. Here is more diffuse, there is some in the nucleus, and it's not working well. So our idea is that what happens is that this splicing process is very peculiar and in some way gets stuck in an aborted spliceosome when the nuclear system that quality control the messengers and see pre-mRNA partially spliced or in the process of splice degrades it in the nucleus. So this one never gets out. A little bit gets through and is spliced and gets out. While the cDNA has no splicing problems and gets out and produces protein the normal amount. We still don't know which is the molecular process at this stage. It could be something at the level of nuclear transportation. It could be something at the level of other markers that the splicing process puts there. And this is work in progress. So, as I said, we were happily working in TDP-43 alone. Nobody in the world cared about it until this paper came out. And Virginia Lee group discovered that these aggregates in the brain of patients were ubiquitinated and phosphorylated TDP-43. From the beginning, we were attracted to this feature. Nucleus, it was empty of TDP-43 staining. It's not always like that, but even the authors in that paper noted it. And we thought that knowing TDP-43 from a long time, we thought, well, if there is no TDP-43 in the nucleus, the cell must be suffering a lot. So we thought that that was the problem, so we bet for a loss of function effect due to capture of TDP-43 in the cytoplasm. And first of all, and you'll hear more later, we did a test in Drosophila, saying, well, if we remove TDP-43 from Drosophila, what happens? And Drosophila that does not have the TBPH, actually, becomes paralytic. So develops, is normal. Note that the eye structure is normal as well. And that's important for it comes later. The eye structure in Drosophila does not seem to be dependent on TDP-43, while instead the neuromuscular junction, it does. This is the normal one, and this is the knockout one. The number of synaptic buttons are less, the structure is more deformed, and Fabian will tell you a lot more about that in a talk later this morning. So what I am going to talk to you now is, well, what about these aggregates, and can we mimic them? What is causing them? And a few years ago, 5 years ago, 6, when Andrea D'Ambrosio was young, he was a PhD in my group, and he did this experiment where we were trying to map. We were attracted immediately about this region of the C-terminus with a series of Q and N, glutamine and asparagine amino acids. And we did a peptide and competed in a gel shift experiment in an HRMPA2 complex to see if there was an interaction that was in some way interrupted, disturbed by this peptide. And in fact, this is TDP-43 bound to its target UG. This is TDP-43 plus A2. So this is the complex A2 TDP-GU. And this is the situation when we add to that mixture a peptide that goes from 330 to 360, more or less, that contain this region. A scrambled peptide doesn't make any effect. So what it was funny was not only that this A2 TDP-43 complex was disrupted and disappeared, but that there was this accumulation of heavy material in the origin of the gel. And we thought this could be aggregates of some sort. So we tried to mimic that in cells, and we couldn't until a postdoc realized that one copy was not enough. If you repeat that sequence, 331, 369, more than 4 times, and you link it to GFP, you get aggregation in the cell. This aggregation is triggered by the, we call it, and I will keep calling it during the talk, 12QN, 12 times repetition of the QN-rich segment. It's triggered by that. They are phosphorylated and ubiquinated. You can see endogenous TDP-43 colocalizing with them. They are not toxic as far as LDH assay in cells is concerned, but there is no change in TDP-43 function. If you go and look at genes that need TDP-43 to splice, they are working fine. If you transfect CFTR exon 9, they work fine. And that's consistent with an inefficient capture of TDP-43. So we say, why is inefficient? So we thought that there may be something else in the molecule, in TDP-43, that helps this capture by the aggregates. So, well, this was to show that in extreme case of the 12QN, we have some nuclear emptying, but not very usually. This is the colocalization with endogenous, and this shows that when you have RNA binding mutants, you have aggregates all the same. When you have a deletion of the 330, 360 of TDP in the transgene, that doesn't colocalize with the aggregates. So, but anyway, coming back to the story, what else is needed? We link the 12QN to the whole TDP-43. So all the molecule intact, and this region repeated 12 times in the same molecule. And at this stage, we got beautiful aggregates that are completely cytoplasmic. I don't know if you can see, but with big holes in the place where the nucleus was. So that means that TDP-43 is being sequestered in the cytoplasm away from the nucleus. If you transfect this molecule into neuronal-derived cells, you get aggregates as well. And very nicely, you can see them also when you differentiate them along the dendrites or axons or whatever are making in culture. So that was very exciting because the splicing was changing as well. This is an endogenous gene, polydip3, that is regulated by TDP-43, is very well known, has been confirmed by several papers and from different labs. And you can see that the effect of SI TDP is to make only, sorry if not very visible, this variant is the shorter variant with this splicing done, and this is the longer one. If you remove TDP, this splicing is activated, and this is the only variant. And you can see the shift with SI. Same shift and better gel you have here. So when you have the aggregates, it's exactly equivalent to that. Only the short form of the protein is present. This is the messenger, and actually this is the protein. So these aggregates are affecting splicing. The same is valid for the CFTR exon 9 test. I mean, here you have the SI, and when you have the aggregates, the situation is identical to the SI. And this is the polydip again. So we have now a system that produces aggregates and is acute, let's say, in producing splicing effects. I would say now that don't forget the GFP 12QN, when there is missing some part of the molecule, is not functional in splicing in the 48-hour assay that we do in cells. But you'll see later that if you put it in an organism, it may be is producing effects a week or two later. Well, what the aggregates do is very simple. They shift from the soluble fraction to the insoluble fraction all the TDP-43 that is in the cell. And this is not a problem of overexpression, because if you overexpress a TDP that doesn't bind itself, so RNA mutant, so no effect on self-regulation, but doesn't have the aggregation peptides, you get normal amounts soluble and insoluble. So what is in the TDP-43 molecule that is causing this enhanced efficiency in sequestering? So we map it, and Valentina Romano did this during her PhD. Now, obviously, as all my postdoc get jobs, she's passing the microphone around. That's the only job that we could find for her. Poor Valentina. This one produce a splice effect, this one as well. We screen it by the polydip2, endogenous gene effect. And you can see here that when you have this species, you get change of splicing, RNA mutant, RNA binding mutant also. But when you delete the amino terminus, all the RRMs, and a good bit of the C-terminal, as long as you leave the 12QN, this molecule does not produce aggregation, but this one does. So this bit seems to be critical. You can see here, it doesn't move, although there are aggregates. So remember that. It's not that that protein doesn't produce aggregation. This one. Produce aggregation, but does not sequester TDP-43. So, did you take the time? Because I don't have clock. 10 minutes. Okay. Let's accelerate. Well, here is the same story. Solubility. So this protein, it makes it insoluble, but not so insoluble, the endogenous one. Our current model is this one, so there is a point of interaction that we have not yet mapped on the amino terminus of TDP-43, particularly that interacts with part of the molecule, with endogenous molecule, sequestering it to the aggregates. You may have aggregates, but if you don't have this region in the inducer of aggregation, you don't get an efficient capture. So you get some, but not all the endogenous in. So we have more or less discarded that these aggregates, at least, the ones that we see with this system, are toxic. So could the aggregation in the general process, in the general pathogenesis of TDP, be protective, or they produce just nuclear depletion and they go away? So to test that, we decided to use a tissue. You remember that we said in Drosophila, the eye structure develops nicely. However, if you use this GMR driver, you get expression only in the eye retinal tissue. Well, this is to do the solubility protocol. If you express GFP12QN in the eye, so the old type of aggregates, those that are not very efficient, there is no toxicity. The eye structure develops well. If you instead overexpress the Drosophila equivalent of TDP, there is necrosis of the eye, there is neurodegeneration. But if you co-express GFP12QN, produce aggregates, and excess of TBPH, then you get a nice eye again. And that obviously we interpret that the 12QN aggregates are mopping up all the excess TBPH and allowing the structure of the eye to develop. This is a specific, and if you co-express GFP and not GFP12QN, the eye remains necrotic, and this is not because the amount of TBPH falls. It's because it's sequestered in an insoluble, non-functional form. Because here you have the levels, and they are comparable between this eye and this eye. The C-terminal region is essential for the rescue. If you co-express TBPH delta C, produces necrosis, but is not rescued by the 12QN, because obviously loses one of the interacting areas. I have gone backwards. Well, this is just to show that it's all a question of insolubility, and that there is no soluble protein anymore when you express the aggregates. This, you have a series of high molecular species as well. Just to show you a bit of the colocalization in the retinal tissue, and that tells you the same thing. There is aggregation, endogenous transgenic TBPH colocalized with the aggregates. It's the same story about this and solubility. So, do the rescue flies see? Or it's just the structure of the eye? So are the neurons functional? And there is this test in the literature that is quite simple, a light and a dark side in a white tube. You put 50 flies one at a time here, and you see, first of all, obviously you test mobility, climbing. If they can climb, if they are comparable climbing, you put them here and see if they go one way or the other, and they are attracted to the light, and you can see that the rescue eye with the 12QN, although overexpression of TBPH, see perfectly well. Obviously, the necrotic eye doesn't see anything. And this is the control of climbing. So the mechanism I already told you. We think that the excess TBPH here is captured by the aggregates and inactivated. Obviously, this will, if TDP was essential for eye function, you should expect a non-functional eye, but TBPH, as we show in the knockout, is not essential for that. So, at this point, aggregates are protective. That is not what we see in the patients. So we kept doing the same thing, expression of GFP12QN in the fly, but pan-neuronal, so in all the neurons. And there we have, with the ELAF driver, we have expression. And what is nice is that this system shows a feature similar to the disease in humans. At the beginning, the fly that is producing aggregates is okay. And only after 1 week or 2, if you are working at lower temperature, you get a drop in mobility. And you have a shortening of the life. So in this case, aggregates are harmful. And they are causing a loss of function. That is obvious, no? In the eye, the loss of function is not important. In the neuromuscular junction is. But this was just to show you that where is the mouse? That the effect of the climbing is quite dramatic. The tubes on the right are normal flies. The tubes on the left are those expressing GFP 12QN.
Bye-bye.
Ah, with sounds. This is new. You can see these ones are climbing fast. You count those here. These ones are almost immobile except few.
Perché sono le 5 linee.
Chi è Raffaella? Okay. I could finish here. I just wanted to tell you that these aggregates have transient, at least in transient infection, they display autophagosomal markers. So we thought that there was a possibility for approaching a therapy, and a general model explained like this. Normally, you have the shuttling of the protein comes out of the nucleus and goes to the cytoplasm, but is transported immediately backwards, and this is a normal function. At a certain point, because aging, because any other stressful effect, it starts to accumulate in the cytoplasm. This nuclei starts to be lacking TDP, so it will produce more because of the autoregulation process. The signal is there is not enough TDP. So it makes more that goes out and binds to the aggregates. The nucleus keeps doing more until its capacity is overcome by the capacity of the aggregates to capture TDP. At this point, you have the pathology. So first step, aggregation is protective, and if your autophagosomes are working well, is wipe it out immediately. Something goes wrong with autophagocytosis or with TDP production, and then you get, you start to get this situation until a critical situation where you don't have any more TDP-43 in the nucleus. So this is the model of how it works. We think that one can interfere with it. I am not going to bother because we don't have much data on this, but there are some compounds that do work in dissolving the aggregates. I don't know if for a patient will be good or bad. We'll see how good it is for the flies, but we are doing that. And I want to thank all the people in the lab. Jeremias has done most of the work on the self-regulation, and then Valentina, the mapping of the TDP-43, and Lucia, Rafaela, helped by Fabian, have done, Natasha as well, most of the work on the flies. And Emanuele has supervised a little bit everything. Thank you very much.